So what does looking at linked genes actually mean? So we've spoken about how linked genes are going to appear on the same chromosome as each other. And in the example that we're looking at on the screen here, if I can just get you to focus on the two chromosomes that I'm highlighting now in purple, okay, we have an example of the A gene and the C gene. And we can see that this individual has a homologous pair of chromosomes that have the A and the C gene on the same chromosome. Okay, so this individual we would say is heterozygous for the A gene. They have a capital A, lowercase a for that particular gene, so heterozygous. And if we look at the C gene that is across that homologous pair of chromosomes, they're also heterozygous for that particular trait. We can see that they've got the capital C and the lowercase c. Okay, so when we look at linked genes, the gamete combination that's produced and then passed on to future offspring can be a little bit unusual. And what I mean by unusual is we can obviously pass on our chromosomes as they are to our offspring. So for example, if we're looking at this individual here, this individual might pass on the blue chromosome that they have to their future offspring, and they would inherit a capital A and a capital C. Or they might pass on the red chromosome to their offspring and get the lowercase a and the lowercase c. What can also happen with linked genes is we know that in a homologous pair, when gametes are forming or during the process of meiosis, crossing over can occur. And when crossing over occurs in prophase 1, it means that part of this red chromosome here might swap with part of the blue chromosome in that homologous pair. And then we end up with a different combination on each of those homologous chromosomes within the pair. So that's what this image is trying to show you here. The beginning of the image shows you the homologous pair as it appears in the parent, okay, just here as I've been describing to you. And then as we move down the image, we're actually seeing what happens to this homologous pair of chromosomes as it goes through the process of meiosis, specifically interphase here, where we're seeing the chromosomes double up and become sister chromatids. And then these two images down here are showing what happens when crossing over is occurring and we get a little bit of that DNA swapping. So we can see that with the color change and also with the alleles being flipped with the capital C and the lowercase c. You then move into the middle of the image here, which is then showing you what would result in each of the 4 gametes that this individual would produce. Okay, so we can see that the original blue chromosome at the top of this set of sister chromatids is appearing up the top here. The bottom part of that sister chromatid has appeared there. The top of this sister chromatid is appearing here, and then the bottom of this one that is completely red is appearing all the way back up here. Okay, so this is just trying to show you that when we're looking at linked genes, you can potentially pass on the original formation of your chromosomes, so completely blue as appears here, or completely red as appears here, and they are known as parental gametes. Okay, they are the most likely combination that a parent is going to pass on to their offspring because that's how their chromosomes naturally occur. The other two combinations, where you're seeing mostly red and a little bit of blue here, and then mostly blue and a little bit of red here, those gametes are known as the recombinant gametes. Okay, and they form because crossing over has occurred and we see some of those alleles switching places, so they're no longer on the original chromosome that they appeared. They are less likely to be passed on to any offspring because the only way that they can be passed on to offspring is if crossing over occurs. Now we know that crossing over is a chance event, so it doesn't always occur, and there is the potential that those chromosomes will stay in their original formation and be passed on just as they are. So when we're looking at linked genes, we also need to think about the way that we are labeling or writing down the genotype for these individuals when we are looking at linked genes. So typically when we were looking at independent genes, if we were looking at an individual that was heterozygous for both traits, we would have written capital A, lowercase a for the first trait, being heterozygous, and then capital C, lowercase c for the second trait. And we would have written them right next to each other in a straight line. So when we see genotypes like that, we assume that they are unlinked or assorted independently, not on the same chromosome. when we're looking at linked genes, we tend to write them using, I guess, a fraction format. And that fraction format, you can see some examples of it here on the right of the screen, is to help us identify what allele combination is appearing on each of the 2 chromosomes that this individual has. So for example, if we look at the homologous pair of chromosomes on the screen here, we can see that our first chromosome, the one in blue, the allele combination on this chromosome is a capital A and a capital C. So we would write that combination on one side of our fraction. And we can see in the examples on the screen here, they've done that on the side of the / or above the denominator if we're doing it horizontally. Your second chromosome, the one in red, has the allele combinations lowercase a and lowercase c. So again, you need to be writing that on the other side of your fraction. So lowercase a, lowercase c here, if you're using the / or the lowercase a, lowercase c at the bottom of the fraction there. So as long as you're using some way to denote which chromosome has which alleles, it doesn't really matter what order you write your fraction in. Okay, so I could just as easily write my fraction as lowercase a, lowercase c over capital A, capital C, because it is telling the reader that on one chromosome I'm going to find the lowercase a and lowercase c alleles, the ones in red, and on the other chromosome I'm going to find the capital A, capital C alleles, the one in blue. So now we're going to have a look at an example of a dihybrid test cross, but this time we're looking at a specific example where we know these genes are linked. And the reason we know they're linked is 1, I'm telling you, but 2, we're going to have a look at the phenotypic ratio that's produced, and it will not be a 1:1:1:1 ratio. And again, if it's not a 1:1:1 ratio, that tells us that we're not looking at an example. We are looking at an example of linked genes. So our scenario is we've got Sarah, a heterozygous here. So we're told Sarah is heterozygous for 2 genes, elliptical blood cells and rhesus positive, and Dave is a homozygous recessive for these 2 genes. In this case, we can write the genotypes as follows. And the reason that we can write the genotype as follows is we have been given images of both Sarah and Dave's chromosomes so that we can see their specific allele combinations on each of those chromosomes. So for Sarah, we can see that she's got a capital D and a lowercase e allele on one chromosome. So we can write that on the top of our fraction here, or one side of our fraction, and we can write the lowercase d, lowercase e on the other side of our fraction for Sarah, and that gives us her genotype. So she's still heterozygous because she's got a capital letter for each of those genes and a lowercase letter for each of the genes as well. Whereas Dave, because Dave is homozygous recessive, both of his chromosomes are going to have exactly the same allele combinations. So the lowercase d, lowercase e on one side of the fraction, and the exact same combination on the other side of the fraction, because both of his chromosomes will have the same allele combination. From those genotypes, we can then work out the gamete combinations that each parent is going to be able to pass on to their future offspring. Now Dave's pretty easy because Dave is a homozygous recessive for both traits. The only thing that Dave is able to pass on to his children, regardless of whether crossing over happens or not to produce the recombinants, Dave can only give his children a lowercase d, lowercase e, or he could give this lowercase d, lowercase e. Even if crossing over did happen and this lowercase d swapped with that lowercase d, he's still going to pass on a lowercase d and a lowercase e. So he can only pass on the lowercase d, lowercase e combination to his children. Sarah, on the other hand, because she is a heterozygous for both traits, she can definitely pass on her red chromosome as it is, so capital D, capital E, and that would be a parental combination. She can pass on her green chromosome as is, a lowercase d, lowercase e, another parental combination, or if when Sarah's gametes are forming, they do undergo crossing over during prophase 1, she might produce a mostly red chromosome, but the capital D and the lowercase d alleles have switched. So she might end up producing a mostly red chromosome with the lowercase d from the green chromosome over here, producing our lowercase d capital E combination, or she might pass on majority green chromosome with that capital D now sitting on the top of the lowercase e chromosome, giving this combination here. So Sarah can actually produce 4 different gametes, whereas Dave can only produce 1. So if we were to draw that up in a Punnett square, this is what the Punnett square would look like. Obviously, Dave can only give that one combination here on the side, and we've got Sarah's gamete combinations along the top. So both of these were the parentals, the original combinations on her chromosomes, and the last 2 combinations at the top were the recombinants. So they would only be produced if crossing over occurred during prophase 1. Okay, we can then see I guess the potential genotype and phenotype combinations of the offspring being shown in the Punnett square below. So you just fill it out as normal, just making sure that you keep your alleles on either side of the fraction as they appear. So we can see here the capital D, capital E was inherited from Sarah from above, so that's on one side of the fraction, and the lowercase d, lowercase e was inherited from Dave, so it's on the other side of the fraction, just to be consistent when we're writing it. So what might that look like? Well, if we're talking about phenotypes, if we refer back to that first slide that I gave you when I introduced Dave and Sarah, these are the possible phenotypes that we're going to produce. So this first combination here, we've got our phenotype in yellow. Our second combination here would be the phenotype specified in red. The third combination is the phenotype in green. And then the fourth combination is the phenotype specified in purple. Okay, remember our yellow and red combinations are our parental combinations, so they are what will be produced when crossing over does not occur in Sarah, and it is more likely to see these particular phenotypes in the offspring than the green and the purple, because the green and the purple would only be produced if crossing over occurred in Sarah's gamete formation process or in meiosis. They are the recombinants and they're less likely to occur. So we would expect to see a higher number of individuals having these 2 phenotypes and a fewer number or a lower number of individuals seeing these 2 phenotypes. So let's have a look at a real world scenario in terms of what numbers would actually be produced for Dave and Sarah if they were to reproduce. And this is, I guess, our real world example. So we can see Sarah's gamete combinations along the top there, Dave's down the side, and then we've got our phenotypic and genotypic outcomes listed in the middle of the table there. So this is just another representation of a Punnett square. I guess the extra bit of information that's in this Punnett square is the numerical value that's been added to each of those genotypes and phenotypes. So looking at the numerical value here, this is trying to give us, I guess, the percentage representation of each of those genotypes and phenotypes. So for the first 2, the parental combinations, it was a 49% chance of producing either of those. Whereas the recombinant combinations, the ones on the right, they're our 1% chance of producing those. Okay, so it is very, very unlikely that Sarah would undergo crossing over where the D and the E allele would be able to swap or form those recombinant gametes, a 1% chance for each of them, or 2% in total, and it is highly likely that if Sarah and Dave were to reproduce, that they're probably going to produce an offspring that has the parental combinations from Sarah. So again, if we look at those phenotypic and genotypic ratios, or the percentages of the potential offspring that are being produced, we can see that it's 49 to 49 to 1 to 1, which is very, very far from 1 to 1 to 1 to 1, or very, very far from 25%, 25%, 25%, 25%. So looking at that information that's being produced in this Punnett Square that we're looking at the screen on the screen here, we can say with 100% certainty that the two genes that we're looking at that Sarah and Dave are trying to track through the generations here, so the D gene and the E gene, these are 100% linked genes because they are not producing a 1-to-1-to-1-to-1 ratio when we perform a dihybrid test cross.